17 and 18, not my two favorite chapters, but let's get going. I was just outside helping my wife start the lawnmower. It's not that she couldn't start it, it's a brand new lawnmower and we haven't used it yet. Or she hasn't used it yet, so she didn't know where the choke was or where the blade is or how to reverse it. It's a Cub Cadet, so our other lawnmower is a John Deere. Expensive. Wow, John Deere's are ridiculously expensive. She's in the West Orchard. We have our orchard split in half. Um, about half of it is east of the house, and the rest of it's south of the house. And the apple trees are starting to blossom, so it's kind of exciting. We haven't seen any bees yet. Maybe in the future. Hopefully, otherwise we won't have any apples. Okay, there we go. Okay. Chapter 17, Learning and Memory Biological Perspective. Not my favorite chapter. This is actually the second time I've started this lecture. The last time I fumbled over the first part. As humans, learning how to get along in the society that we have created takes a lifetime. And actually, I was just reading a thesis, and that's exactly what she's talking about in her thesis. The Navajo people needing to figure out how to get along in the Navajo society. Our memory is the culmination of our ability to learn about life and life circumstances. It is our memory that allows us to act appropriately in situations. If we kill someone or drive on the left side of the road in the United States, if you drive on the left side of the road, you're okay in Japan, you're okay, you're okay in New Zealand. I think you're okay in Australia too, and of course in England. We will be taken out of society either through death or incarceration if you drive on the wrong side of the road. And you got to be careful. And I've lived in both, I've driven in both England and Japan. So you got to be careful. Our memory dictates our behavior. Oddly, psychologists and medical doctors have been able to trace and map the memory process using individuals who have damaged select areas of their brain due to tumors, lesions, or trauma. These insults to the brain cause lapses of the memory that looked at in toto, construct a memory map to the process. Skill learning is learning to perform a task that requires motor coordination. That's skill learning. Priming is exposure to stimulus which facilitates subsequent responses to the same or similar stimulus. Conditioning is an individual comes to associate two stimuli or its stimulus and response. Edward Tolman has hypothesized through research with rats that they create a cognitive map in their minds from their experiences. And this is the maze that he used to see if the rats could remember things. Of course, he's not with us anymore. Tolman also was able to prove that rats had knowledge that, unless tested, could not be discounted. This knowledge he referred to as latent learning. So a lot of times you learn things. I mean, I may tell you a story, and 5 years from now you go, of course, you've never thought about it, you've never used it before, and then 5 years from now you need it, and all of a sudden it's there. It's in your memory. Tolman later, and this is known as latent learning. There's a lot of latent learning that takes place. You walk into a room and there's something on the wall that you don't really look at, you don't really concentrate on, and then when somebody asks you, "Was there a picture in there?" You go, "Yeah, it was a picture of an old man." You didn't really look at it. You didn't really think about it. You didn't put it in long-term memory, but it was there. And that's known as latent learning. Tolman later wrote that different kinds of learning may follow different laws. One of the things that we have noticed is that now that we have cell phones, people are concentrating on their cell phones, and they're not experiencing their environment around them. Let me give you a quick example. We were driving home from soccer practice, my grandson and I, and we were headed toward Clinton, and right in front of Clinton is a chemical plant, and they had an explosion. And as we're driving down the road toward the chemical plant, there's this huge explosion. Well, we couldn't hear the explosion; it was too far away, but I could see it, and there the sky lit up. This is at night. The sky lit up, and by golly, he didn't see it, and of course, we didn't hear it. There wasn't anything to hear. Well, there was eventually, but we were that far away, and he didn't experience it at all because he wasn't paying attention. He was paying attention to his cell phone, even though the sky just lit up with light because of the explosion. Don't worry, nobody was hurt. They said it was a controlled explosion. We believed him. Tolman later wrote that different kinds of learning may follow different laws, and he didn't notice it because he was so concentrating so much on his cell phone. And that's one of the things that we're seeing. People are walking into each other. People are not paying attention. They have no idea who's around. And it may be one of the reasons why school shooters are shooting so many people because nobody's noticing. People are concentrating on their phones and they don't notice anything. The only way to tell them that something horrible is happening is by putting it on their phones. Iconic memory, brief memory of an image, part of the short-term memory. Echoic memory, of course, is brief memory of a sound, part of the short-term memory. And short-term memory, iconic, echoic, or cognitive memory that lasts for about 30 seconds if unrehearsed. And the rehearsal has to do with thinking about it. You can put things in short-term memory just by thinking about them, and they will go into an intermediate memory so that it stays there for an extended length of time if you think about it. And that's one of the reasons why my grandson, he wasn't paying attention was his problem. Some psychologists refer to memories that may last longer than the short term, an hour or a day, but are not put in long-term memory as intermediate term memories, and that's usually that's what I was just talking about. Long-term memory is permanent memory, memories that last the rest of an individual's life. It's in there somewhere, you just got to find it. Use the right cues. The capacities of long-term memories are almost limitless. Research shows that college students can recognize 50,000 words. People who learn a second language acquire 100,000 items of knowledge. Memory trace is the path that memories are placed in the long-term memory, and that is you've got to find that memory trace. You got to find the right cue in order to bring it out, all that information. Now, you may not know this, or I may have bragged about it over and over and over again, but I have I have 5 degrees. 2 of them are in psychology. One is in political science. One is in, wait a minute, one's in English and one's in medical technology. Okay, so that's 5 disciplines, or that's actually 4 disciplines that I have education in. And I've got lots of information in my brain somewhere. It's just floating around in there. And but the question is, how do I get it out? And actually, I read in all 4 of those disciplines. I have, and plus other areas. I'm very interested in history. I'm very interested in archaeology. I'm very interested in natural history, and I get National Geographic every month. And you never know what's going to be in there. So, yeah, I've got lots of information in my brain, and the question is, how do you get it out? Or how do you find it? You studied this, or you studied something. How do you find it in your brain? Or you even read about it. How do you find it in your brain? And of course, this irritates my wife to no end because she can't tell me anything because I just read an article about it. It really irritates her. Really irritates her. And we've been married for 46 years, I think. Wait a minute. We got married in 79. Yeah, this is the 46th year. The more severe the damage to the medial temporal lobe, the more impaired the ability to establish new memories. Work with people with hippocampal damage shows that the hippocampus is probably the final location of memory formation. These individuals can still support some degree of cortex memory formation, but only certain types of memory. One system includes the hippocampus. This system acquires information about relationships among stimuli and events, and this is known as declarative memories. And there's your hippocampus. As you can see, it's right in the middle of your brain. It's down a little bit lower. If you press on your temple, then directly in from where you're pressing is your hippocampus. It's not close to the surface. It's right in the middle. It's protected by the rest of the brain mass. There is a system that includes the dorsal striatum. And there's the dorsal striatum right there. This system mediates the formation of reinforced stimulus response associations, habits, or non-declarative memories. There's a system that includes the amygdala. This system mediates rapid acquisition of behaviors on the basis of biologically significant events with effective properties, events that arouse your emotions. Anything that arouses your emotions, it's going to go into your memory. And the reason is because if something aroused your emotions, it means it was either dangerous or is happy. It was something that is important to you. That's why your emotions were aroused. Researchers have found that animals who catch food have neurons in the hippocampus that are activated when they get into a particular location where they have cached food. These neurons are referred to as place cells. These special view cells are seen in the larger hippocampuses of birds who catch food, as well as squirrels, as well as any animal that they catch as food. This is really important because this keeps them alive. So they need to remember where their food is. When information is properly encoded, there are two areas of the brain that are activated when the memory is remembered: prefrontal cortex and the parahippocampal cortex. Pictures are remembered in the right prefrontal cortex and both hemispheres of the parahippocampal cortex. Words are remembered in the left prefrontal cortex and the left hemisphere of the parahippocampal cortex. And that's what we're talking about right here. The hippocampal system is not a repository of long-term memory. Permanent storage is thought to occur in regions of the neocortex, where the information is first processed and held in short-term memory. This information is further processed in the medial temporal region, but this area is not the location of long-term memory either, merely an area of consolidation. And if you've ever walked into a room and couldn't remember you couldn't remember why you walked into that room. This happens to me all the time. My wife tells me I'm an absent-minded professor, but yeah, that's the problem. You don't have the right cues. You thought of, oh, I need to walk into this room in another room. Usually, if you walk back to where you were when you had the memory the first time, or when you were thinking this is what I need to do, usually you can remember why you walked into the whatever. Usually it's the kitchen. I can't remember why I walked in. Imaging research has verified two regions of the brain used in establishing declarative memory: the medial temporal lobe and the diencephalic system. Imaging of the brain shows activation in both encoding of new material and retrieval in that system. And there you go. Encoding takes place in the rostral regions of the hippocampal area. Retrieving episodic memories are located primarily in the caudal portions of the hippocampus. Encoding takes place in the rostral regions of the hippocampal area. Retrieving episodic memories are located primarily in the caudal portions of the hippocampus. And there you go. Looking at the models of encoding and retrieval of the same information, researchers and theorists ponder if the two systems aren't identical. And this is known as memory trace. There are three types of skill memory that researchers have delved into to discover brain areas involved in them: the sensory motor skills, perceptual skills, and cognitive skills. Imaging of people performing skills from memory show that there are three areas activated in this process: the basal ganglia, the cerebellum, and the motor neocortex. Okay, so what are we talking about here? We're talking about, this is the motor neocortex is right up here. This is the basal ganglia, right in the middle of your brain, and there's your cerebellum. Now, we used to think the cerebellum only had to do with movement, but now we know that it is activated, especially if it is a memory that involves movement. Like why I walked into the kitchen. This would go through my cerebellum, and this is how you retrieve information. It has to go through all three areas. So in essence, when you are thinking about why in the world did I walk into the kitchen, it's your neocortex that is being activated. Since you're walking in there and it had something to do with walking into the kitchen, your cerebellum is activated. And of course, all of this comes out of the basal ganglia. Amnesia is a severe memory impairment, but seldom complete memory impairment. Retrograde amnesia is back in time from the incident. You can't remember some or all of one's past after a select incident. Anterograde amnesia is forward in time from the incident. You can't establish some or any new memories after an accident. So retrograde amnesia is what you see on television all the time. It's a real good plot twist where the person loses their identity because they can't remember who they are. Anterograde amnesia means you can't establish new memories, which is kind of interesting. The interesting thing is that you can remember your past, you just can't make new memories. And sometimes blows to the head, traumatic brain injuries will cause either of these amnesias. Usually they're short-lived. They don't last for very long, and usually your memory comes back. It usually has to do with a swelling in a certain portion of your brain. As soon as that swelling goes down, of course, your brain's trying to protect itself all the time, and if you've had some kind of damage that has been done, that portion of your brain may swell, and if it swells up to protect itself, that may have something to do with memory retrieval, and that's why you have either retrograde or anterograde amnesia. When H.M. was 16, he developed seizures. By age 27, the seizures were so bad that he had to quit work. Medication could no longer control his problem. A neurosurgeon determined that both temporal lobes were involved. He excised most of the anterior temporal lobe, the amygdala, and the hippocampus in both hemispheres of his brain to stop the seizures. H.M. can't remember anything from 10 years before his operation, and this is known as retrograde amnesia. H.M. only has short-term memory and can't establish any new long-term memories. This is anterograde amnesia. H.M. has now died. We know this, and this is actually a picture of him, I think. Yeah, he died like last year. It was really kind of interesting because nobody knew who he actually was. There are two types of memory: declarative memory, which represents information acquired by learning. These are the facts that you have in your brain. And procedural memory, things that people know that they can show by doing. HM could not form new declarative memories, but he could form new procedural memories. So if it was something he had to do, he could figure that out. And these are the areas that we're talking about: the cerebellum, the basal ganglia, and the hippocampus and the neocortex. There you go. After HM's case became widely known, cases of similar results came to the attention of the medical community. Herpes simplex viruses can, on rare occasions, destroy tissue of the medial temporal lobe, causing similar results. Ischemia, reduced blood flow to the brain, as in the case of a stroke or heart attack, can damage the medial temporal lobe, causing similar results. Encephalitis, inflammation of the brain, causes similar results. And it's encephalitis that is this is the problem that people have when they have measles. They get a high temperature that destroys. They get in some that creates encephalitis that destroys portions of their brain. And this is one of the reasons why measles is so dangerous. Usually, if you can get over it fast enough and your temperature is not too high, you don't have to worry about any of this stuff. But some people have lingering the measles, their fever, they're more sensitive to fever, and it destroys portions of their brain due to encephalitis. NA was messing around with a miniature foil, which is used in fencing. These guys are using a miniature foil. They're not very long. Somehow the sword entered his nostril and skewered him through his sinus cavities and into his diencephalon. The injury damaged the dorsomedial thalamus, destroyed the mammillary body, and created a lesion on the hypothalamus that destroyed 2/3 of the organ. NA suffered an anterograde amnesia. He suffered anterograde amnesia. NA's short-term memory was intact, but his declarative memory was absent, though he had procedural memory. Korsakoff syndrome is a profound amnesia associated with chronic use of alcohol. There is deterioration in the diencephalon, the hippocampus, the amygdala, and some to frontal cortex. The problem is that the liver is so busy processing alcohol, it doesn't do a proper job processing vitamins, especially B1, also known as thiamine. Chronic alcoholics will often display a marked retrograde amnesia. Curiously, alcoholics will often fill in the missing information with fantastic stories that are either constructed from their stuporous, inebriated state or suggested by someone else. This fantasy memory is referred to as confabulation. And for some reason, this happens to alcoholics. We're not exactly sure why them specifically they have this confabulation, but they're never at a loss for words, and you can always get them to tell you a story about something, even though they don't remember. They don't remember anything, and if somebody suggests to them, it may have been aliens down by the creek, they'll repeat that they saw aliens down by the creek. The alien part will stick, but they have no idea what they did because they're used to not having any memories of the past. So they'll just accept whatever memories people come up with. Work by Estonian scientist Ingvar Tulving led to the bifurcation of declarative memory, facts, and information into two types: semantic memory and episodic memory. Semantic memory is generalized information. Episodic memory is autobiographical memory that makes up an individual's personal history. So those are both declarative memories. Casey sustained brain injury due to a traffic accident. After the accident, Casey suffered damage to the frontal parietal cerebral cortex, damage to the right parietal occipital cerebral cortex, severe shrinkage of the hippocampus, severe shrinkage of the parahippocampal cortex. He had a fractured skull, as you can see. Fractured skull is a break in the skull, and a lot of times it causes real serious problems, like what happened to KC. Due to the damage, Casey lost his ability to recall episodic or autobiographical memories. Casey still maintains semantic memory but can't remember who he is. The problem seems to be in the damage to his cortex, not the damage to the hippocampus. Researchers feel that it is the frontal parietal damage that causes the loss of memory. And this is one of the things that we do when we're looking at a concussion. If somebody comes, a football player, gets his bell rung, that's a concussion. They take them off the field and they start asking them about things. They want to know how their memory is. They need to know, they want to get them to talk. They need to get them to talk to find out if they're slurring their words like I am right now. They need to know if they can see, so they'll hold up fingers. They'll ask them to identify things. They need to find out how damaged their memory is. They'll ask them questions. What day is it? Who's the president of the United States? Do you know where we are? These kinds of memory, or these things, we're just trying to get them to remember things to find out how much damage there is to their brain. Depending on where they get hit and where the blow is, usually you get a rebound blow. So if you get hit in the front of the head, you're also getting hit in the back of the head because your brain is rebounding inside your skull. And this is one of the reasons that we ask so many questions. And this has to do with concussion protocol. And usually they don't allow them to start participating again in football or in whatever they're playing until their memory is intact. That means that sometimes it can cause minor damage that causes minor swelling, or just a shock to the system, and they will lose their memory for a short period of time. Then all of a sudden it comes back because it was just a shock to the system. It's like punching somebody in the nose, and all of a sudden they. But then as soon as they start thinking about it, everything's back to normal. Memory can be divided into time-structured stages. Iconic and echoic memory is the initial presentation of stimulus, arrives in the brain as a momentary stimulus that can readily disappear. Short-term memory is iconic or echoic memory that the individual seeks to maintain. This may last for a minute or so. Intermediate memory, short-term memory that you're able to hold because of its novel subject or that is readily organized in the brain. For example, a weather report. Long-term memory, shorter duration memory that is placed in a more permanent area of the cerebral cortex, where it may be recalled days, months, or even years after the fact. Permanent memory lasts a lifespan. Long-term memory can be accomplished using several techniques: skill learning challenging tasks over several sessions. And the question comes, how many sessions does it take to remember something? The magic number tends to be 3, but some people have the ability to remember things from the first session, if they're novel enough, or if they're interested enough. If they're not interested, then it takes 3 times. So boring information, if you read it 3 times, or you acknowledge it 3 times in your brain, you think about it 3 times, it's stuck. You got it in there. 3 times. Priming, stimuli that is altered due to previous exposure to the same or similar stimuli. Conditioning, tying a stimulus response to a reward or punishment. Classical conditioning is tying a stimulus response to an unconditioned stimulus. Operant conditioning, tying a stimulus response to a reward or punishment. Research indicates that every time a memory is called up, there is potential for distortion. New memories or new information may change the structure of the memory. The structures of questions or memories can further plant false memories. Some people are quite susceptible to false memories. Stories made up by authorities, usually that will stick. In one study, 25% of the respondents accepted the false memories. The ability of implanting false memories clouds the accuracy of such controversial memory formation as recovered memories of childhood sexual or physical abuse. Delivered testimony that has been acquired from intensive questioning in a criminal trial also has questionable accuracy. And here we go. I think if it's still there, there. Select attention test.
This is a test of selective attention. Count how many times the players wearing white pass the basketball. How many passes did you count? The correct answer is 15 passes. But did you see the gorilla? This video is from research by Daniel Simons and Christopher Chabris and is copyrighted. It is available for use in talks, training, and teaching on DVDs from VizCog Productions. Learn more at theinvisiblegorilla.com.
Ready to tackle your debt? We can
Whoops.
help.
Okay, just a second.
Vision
Sorry.
subscription can
Had to
help you
go
visualize your debt picture, boost your financial IQ, and even submit
Okay, I got
payments
another one
directly
for
through
you.
the app.
Wait a minute. Okay, let's do this one.
This is a test of selective attention. Count every time a new exclamation point comes on screen.
Now it's time for our wrap-up. Let's give it everything we've got. Ready? Begin.
Artificial amateurs aren't it all amazing? Analytically, I assault, animate things. Broken barriers bounded by the bomb beat. Buildings are broken, basically I'm bombarding. Casually create catastrophes, casualties, canceling cats got their canopies collapsing. Detonate a diamond tank daily doing dope. Demonstrations, Don Dada on the down low. Eating other editors with each and every energetic, epileptic episode, elevated etiquette. Furious, fat, fabulous, fantastic. Flurries of funk felt feeding the fanatics. You've got great global goods gone glorious.
Welcome to Intermission. Yeah, intermission.
Now it's time for our wrap-up. Let's give it everything we've got. Ready? Begin.
Artificial amateurs aren't it all amazing? Analytically, I assault, animate things. Broken barriers bounded by the bomb beat. Buildings are broken, basically I'm bombarding. Casually create catastrophes, casualties, canceling cats got their canopies collapsing. Detonate a diamond tank daily doing dope. Demonstrations, Don Dada on the down low. Eating other editors with each and every energetic, epileptic episode, elevated etiquette. Furious, fat, fabulous, fantastic. Flurries of funk felt feeding the fanatics. You've got great global goods gone glorious. Getting godly in this game with the glorious. Hit 'em high, hella hype, historical. Hey, Holocaust hymns, hear 'em holler at your homeboy. Imitators idolize, I intimidate. In an instant, I'll rise in an irate state. Juice on my jams like Jericho's chopping joints.
All right, I think I've got one more. Oh, one more. There we go. This is a commercial, actually. Advertisement, I'm sorry. Come on. All right. There we go. Now, come on. I've been having trouble with YouTube lately. Here, let me try this again. That may not work.
To test just how much attention the attention-stealing design of the new Skoda Fabia actually steals, we left one parked on this ordinary road in West London. We wanted to see if its sharp crystalline shapes, bold lines, and lower, wider profile would attract the desired level of attention. Will the 17-inch black alloy wheels stop passers-by in their tracks? Will the angular headlights attract the attention of other road users? Will a crowd gather to check out its fresh, sporty look? Well, not quite. But did the attention-stealing design distract you from noticing that the entire street has been changing right before your very eyes? Don't believe us? Have another look. Did you spot the van changing to a taxi? How about the scooter changing to a pair of bicycles? Or the lady holding a pig? Let alone the fact that the entire street is now completely different. Didn't think so.
Ta-da! Okay. I hope you saw the gorilla. Memory is formed around the structure of our senses. Attributes of memory include time, space, sensory perceptions, response, cognitive effect of the stimuli. Each attribute seems to utilize different neural structures. From studies of brain imaging and memory, as well as evidence from brain damage due to injury and disease, scientists have been able to map different memory processes. Spatial location takes place in the hippocampus, memory of our own response in the caudate, object recognition, the extrastriate visual cortex. And there you go. Caudate nucleus, as you can see, right in the middle of your brain. When functioning properly, the caudate may allow only a little bit of frustration to travel to the frontal lobes as a reaction to an upsetting situation, such as lima beans for dinner. However, a damaged caudate may leave the gate open for too long, allowing a great deal of frustration to travel to the frontal lobes, which may result in an extremely intense emotional response to things as silly as lima beans for dinner. An undamaged caudate regulates and organizes information received from other areas of the brain, such as the occipital lobe, responsible for vision, and transmits the information to the frontal lobes. The frontal lobes then instruct another area of the brain to carry out a particular task based on the transmitted information. A damaged caudate, on the other hand, may not properly deliver the information to the frontal lobes. The result, unawareness, may mean that particular tasks such as getting a haircut is not carried out. The caudate may work much like a secretary in that it is responsible for regulating and organizing messages or information for the boss. If the secretary gives the boss a stack of unorganized mail and telephone messages, he or she may be unable to manage the incoming messages efficiently and become overwhelmed. Whereas a damaged caudate can lead to slowed thought processing, undamaged caudate enables the frontal lobes to perform quickly and efficiently. Extrastriate visual cortex. There's your extrastriate cortex. You saw where the caudate was, right in the middle of your brain. And of course, the extrastriate visual cortex is in the back of your brain. Memory formation and use involves 3 stages: encoding, iconic memory pictures, right prefrontal cortex, parahippocampal cortex in both hemispheres, and echoic memory, sounds, left prefrontal cortex, left parahippocampal cortex, right on your temporal lobes. Iconic memory, okay, there's where it takes place. You can see where it is taking place. There's the echoic memory right around your ear, I guess where it needs to be. Memory formation and use involves 3 stages. Encoding and consolidation takes time to place information from the intermediate memory to long-term memory. The hippocampal system is part of the process, but not a repository for long-term memories. Memories are further processed in the medial temporal region and probably the midline diencephalic region. Now, long-term memory takes place in various regions of the cortex. Okay, there's the hippocampus. There's where everything's taking place, right up here. And I can't remember why I put that in there. Memory formation and use involves the 3 stages. Encoding, consolidation. Last is retrieval, of course. During retrieval, the area of the cortex that is used for the type of memory is activated. Researchers have discovered that emotion often enhances memory. However, emotion can weaken information that is not central to the emotional theme. Memory seems to be enhanced by the beta-adrenergic stress hormones. Research indicates that emotional stories are easier to learn. This is because stress hormones from the adrenal glands activated by the emotions activate beta-adrenergic receptors. This is why memories of trauma are so readily remembered. To exacerbate the problem for some sufferers of post-traumatic stress disorder, remembering the trauma causes the stress hormones to be activated, stimulation, which in turn reinforces the bad memories to occur again. One hypothesis of PTSD is that since the memory was established by the adrenergic stress hormones, it was remembered better. When this memory is recalled either by intrusive memory or flashbacks, the memory is reintroduced and so it is reinforced. By administering anti-adrenergic drugs soon after the incident, it would reduce the impact of intrusive thoughts and flashbacks. Looking at combat records from Vietnam, researchers determined that individuals who suffered from traumas as a child were more likely to develop PTSD post-combat. Why? Because those avenues that had been created by the childhood trauma were already there. So the individual was primed to establish new traumatic memories. This may be because of individuals who have suffered from trauma as children already established the beta-adrenergic memory pathway. Drugs such as propranolol, which suppresses beta-adrenergic activity, might be used before or directly after the trauma to prevent such pathways from forming, and actually we have proven that propranolol does not work. They tried it in Afghanistan and Iraq, and it was an abysmal failure. Medial temporal or hippocampal and diencephalic systems are needed to form new declarative long-term memories. These areas are activated in both encoding and retrieval of declarative memories. Okay, I do have PTSD. I was in the military for 12 years. I was in on a lot of body recoveries from airplane crashes. And a lot of times, because they were military aircraft, they caught fire and the individual burned. And because of that, I have strong memories of what burning human flesh smells like, because I was the one that had to take charge of the body afterwards. I know it's an ugly job. Somebody's got to do it, and I was lucky. And because of that, certain smells will give me the whim-whams. One of them is that, and this is one of the reasons I can't really come back to campus. One of the smells is the smell. Mutton has a very similar smell to, at least in my memory. Mutton has a very similar smell to human flesh, is burning human flesh. So while I was on the Staley campus, from time to time I would get bad memories would come flooding back. So it's one of the I try to stay away from mutton. I try to stay away from cooking goat or cooking sheep. But that's another reason why I can't come back to campus. And I didn't really know. It's interesting because it smells, and you just ignore smells unless they're important. Somebody farts in front of you as you're walking. You don't really notice it. You may acknowledge it for an instant, but after that, you just don't even think about it. And that's what was happening. Didn't have anything to do with farting. Had to do with passing the cafeteria when they were cooking, mutton stew or whatever. It just kind of it's like getting slapped in the face. It's really kind of weird. Anyway, that's a reason. You can take that. Oh, that's too bad. That kind of stuff. That's all right. That's okay. I'm not coming back in the fall. So, and there's a couple different reasons. One is that I'm 75 years old and I have a heart condition that difficult to take care of in such an isolated place where I wouldn't have any medical treatment for 75 miles. But that's just the way things work. And I won't worry about it. Medial temporal hippocampal and diencephalic systems, I'm living in Iowa. They very rarely cook any mutton here. Greek restaurants, they used to drive me crazy. Where there was a Greek restaurant in Havre, Montana, where I lived. And every time I went past, I get kind of a really odd feeling. And I didn't really understand what it was. But while I was in Saley, other individuals who were suffering from PTSD, we discussed what was going on, and I have to thank Emery Denny for helping me with my PTSD, and I hope I helped him because he has a different. Of course, his trauma is completely different than mine. What are we talking about? Medial temporal, hippocampal, and diencephalic systems are needed to form new declarative long-term memories. These areas are activated in both encoding and retrieval of declarative memories. I grew up in a pork region. This is really kind of interesting because I have a friend who also was around burning human flesh, and pork reminds him of that. And mutton reminds me of that. So it's really kind of interesting. I grew up around pork, and maybe it does. Maybe pork smells more like burning human flesh than mutton. But I didn't grow up around mutton, so mutton is an alien smell to me. And I guess pork was an alien smell to him, and so as far as his memory is, it's pork, and as far as my memory goes, it's mutton. So, just the way things work. Sensory motor skills such as mirror tracing, basal ganglia, cerebellum, and motor cortex. Cognitive skills such as towers of Hanoi and the cerebral cortex. And that's what the Towers of Hanoi is. If you have children, when they were, I don't know, 18 months old, you probably bought them one of these. And they just put the rings on each of the posts. It's known as Towers of Hanoi. Perceptual skills, learning to read mirror reverse text, occipital lobe activation. Oh, this is non-declarative memory. And you can probably read this, despite the fact that it's backwards. Oh, hello there. I didn't see you come in. I was too busy reading off this teleprompter. It does some pretty cool things. You can edit your scripts on the page or paste them here to import. Huh. That's a Q. I don't know. Oh, that's copy. That's a P. Import. Import them from another program. It'll even save it for you if you happen to leave the page. That's some smart thinking. If you're using an iPad or Android tablet, add a shortcut to your home screen for full screen. There you go. Yeah, I kind of stumbled over this one, especially I couldn't import. I couldn't. My brain started flipping the P and thought it was a Q. There appears to be a degree of decline in the ability to learn and remember as humans age, and this guy's probably younger than I am. How much decline depends on heredity, education, and activity levels of the individual. I have lots of education. My parents both lived into their 90s, and I'm very active. I don't know if that is helping my memory. Obviously, it didn't help my reading of the last slide. Actually, the elderly are able to recall memories as long as the memory cues are adequate. And that is the end of that one. Let's go to chapter 18. Learning memory, neural mechanisms in learning memory. Research shows that memory formation can take place in the development of simple neuronal changes all the way to complex neural changes. Memory is only formed when neuronal changes take place. Simple neuronal circuits may receive input from more complex superordinate or modulatory circuits. However, some memory requires more complex neural connections, relatively complex networks called cell assemblies. Many of the changes in neuronal structure can be measured physiologically. These changes may be presynaptic, postsynaptic, or both. Many researchers believe that long-term memory requires significant neuronal structural changes. Donald Hebb suggested that if a neuron frequently stimulated another neuron, that it would make a permanent change in the stimulated neuron. Practice makes perfect. And I told you the magic number is 3. So if we run over that memory 3 times, you'll remember it. And I can promise you, if you're studying something, go through it one time, put it down, then come back, go through it a second time, put it down, come back. The third time, it's in your brain forever. A Hebbian synapse is a synapse that has changed because of frequent stimulation. This means that the synapse is not only active in stimulating other neurons in the area, but that it is frequently stimulated as well. Neurons that fire together wire together. This frequent stimulation follows Hebb's dual trace hypothesis. Research actually done by the authors of this text, dealing with the weight of rats' brains in a standard environment, an impoverished environment, and an enriched environment, proved that rats who were raised in an enriched environment had heavier brains than rats raised in the other two environments. The area of the brain that had the most significant growth was the cerebral cortex, but especially the occipital cortex. Further research has proven that much of the change takes place because of dendritic branching. Dendritic morphology has been shown to be constantly changing. Fine filaments extend from the dendrites. These are called filopodia. If these filopodia make contact with an axon, they may form dendritic spines. And that's what we're looking at here. Filopodia moving, trying to make a new synapse. When a synapse receives repeated strong stimulation, it creates a stable and enduring increase in the effectiveness of the synapse. And this is termed long-term potentiation. This can especially be seen if the neuron experiences tetanus, a flurry of intense and constant electrical stimulation that creates a permanent change in the synapse. The hippocampal formation is formed by two interlocking C-shaped regions: hippocampus, dentate gyrus, subiculum, the hippocampal gyrus. These three structures are divided into four regions: CA1, CA2, CA3, and oddly, CA4. And there they are there, CA1, CA2, CA3, and CA4. NMDA glutamate receptors acts as an antagonist preventing long-term potentiation. But NMDA does not affect LTP that has already been established. AMPA glutamate receptors increase LTP. Activation of NMDA receptors leads to an increase in AMPA receptors. And we're going to see this right now, I think.
The NMDA receptor is one of two main kinds of receptors activated by glutamate. The other main kind of glutamate receptor is called the AMPA receptor. These two receptors, each with distinct physiological properties, often coexist at the same synapse. Shown here is a model synapse in the CA1 region of the hippocampus containing both types of receptors. Weak electrical stimulation of the presynaptic neuron causes the release of glutamate from the axon terminal. This glutamate binds to both AMPA and NMDA receptors. Although both NMDA and AMPA receptors are permeable to sodium and potassium ions, weak stimulation normally activates only the AMPA receptors, resulting in a slight depolarization of the postsynaptic neuron. When glutamate binds to the NMDA receptor at slightly depolarized or resting membrane voltages, very few ions flow through the channel. This low conductance occurs because the pore of the channel is blocked by magnesium ions, which prevents other ions from passing freely through the channel. Under such conditions, the EPSP will be mediated entirely by the AMPA receptors. Given a stimulus of sufficient strength or frequency, AMPA receptors can depolarize the membrane sufficiently to expel the magnesium from the NMDA channel. The NMDA channel now actively responds to glutamate, admitting not only sodium ions, but large amounts of calcium as well. The calcium acts as an important second messenger, activating several intracellular signaling cascades. For example, some calcium binds to calmodulin, and this complex in turn activates several protein kinases, including calcium calmodulin-dependent protein kinase, or CAM kinase. CAM kinase affects AMPA receptors in two ways. First, it phosphorylates AMPA receptors already present in the dendritic spine membrane, thereby increasing their conductance to sodium ions. CAM kinase also promotes movement of AMPA receptors from intracellular stores into the membrane, making more receptors available to stimulate the spine. In addition to these postsynaptic effects, calcium may also facilitate the release of transmitter from the axon terminal via retrograde signals such as nitric oxide. As a result of the increase in the number of AMPA receptors, The response to a stimulus of a given strength will be stronger than it was before the NMDA receptors were activated. In this regard, the synapse is said to be enhanced, and this physiological change is thought to be one of the mechanisms underlying the expression of long-term potentiation, or LTP.
Ta-da! Okay, that's how we remember things. All that stuff has to take place. When glutamate is released in a synapse that has both AMPA receptors and NMDA receptors, the AMPA receptors require only moderate stimulation to be activated, whereas the NMDA receptors require a greater amount of stimulation. We just saw that in the video. This occurs because magnesium blocks the NMDA calcium channels effectively, keeping the NMDA receptors from firing. And that's why magnesium is important, and we also know that calcium is very important to memory. If the AMPA receptors become stimulated enough, it depolarizes the membrane, removing the magnesium and allowing the NMDA to fire. And we just saw this as well. This allows a large amount of calcium to enter the neuron. So the NMDA can only be activated when the cell is depolarized and there is glutamate present. And we just saw this. The large influx of calcium activates protein kinases, enzymes that enables phosphate group, the phosphate group, that's PO4, to attach to a protein molecule. Protein kinase A, pKa, protein kinase C, pKc, calcium calmodulin kinase, CAMK, and tyrosine kinase, TK. CAMK affects AMPA in two ways. Here, let me throw all these alphabets at you. Phosphorylates AMPA receptors, increasing their conductance of sodium and potassium ions. It promotes the movement of AMPA receptors from the interior of the neuron to the surface of the membrane, and we just saw all of this. CAMK leads to the formation of intermediate term memory. PKC leads to the formation of long-term memory. PKA leads to LTP, which in turn helps to establish both long and intermediate term memory. The activated protein kinases not only help to phosphorylize proteins, but they also trigger the synthesis of new proteins. The protein kinases activate CAMP responsive element binding protein, CREB. CREB in turn binds responsive elements. The elements that CREB binds to are nucleotides. Whew. The nucleotides that are affected are in the promoter regions of the DNA adjacent to genes. This changes the rate of transcription of adjacent genes. This in turn alters the growth and differentiation of the neuron. If the protein that recognizes CREB is genetically disrupted, it causes a syndrome that is characterized by intellectual disability. Wow. But there are also CREB antagonists. Inducible CAMP early repressor, ICER, produces 4 proteins that block CREB gene transcription. These proteins block CREB by binding to the genes themselves, but do not promote transcription. If too many ICERs are present, CREB will not induce long-term memory. Another CREB antagonist is phosphatase 1, which inactivates CREB. It is thought that these CREB antagonists exist to pair unwanted memories or to keep them from forming to begin with. The memory process takes place over hours. For the first hour, protein does not have to be synthesized to maintain the memory, but for the memory to form LTP and hence LTM, CREB has to be activated and protein synthesis has to take place. Now we're recognizing all of these it's important to be able to forget things, and I have the ability to forget things, and that's one of the reasons why I didn't suffer from PTSD for the longest time until the memories came flooding back, of course. After 3 hours, select proteins that have been synthesized will decrease, while others must increase to maintain the LTP. At this point, the affected neuron will signal the presynaptic neuron to release more transmitter to maintain the signal. This is known as retrograde signal. Memory retrieval requires finding the proper cue to connect the individual with the neuronal bundle that contains the information. When the memory is found, research demonstrates that the structure of the memory becomes unstable and susceptible to disruption from protein synthesis inhibitors. If there are no changes, the memory will be reconsolidated and return to stable status. This is, of course, necessary for memories dealing with a person's behavior, where inappropriate conduct must be repaired immediately. Epinephrine affects memory formation by influencing the amygdala. It does this by forcing the release of norepinephrine in the amygdala. If the individual takes propranolol, the amygdala's memory-enhancing effects are blocked, and we already know that this doesn't really work. Opioid peptides will also block the release of norepinephrine in the amygdala. And one of the things about propranolol, they thought it would stop PTSD completely, but it doesn't. It blocks some things, but you don't want people to forget everything. And that was the trick. You go into a combat situation and you've got to remember to lower your head, and if you wait for the sound of them loading their gun, you got 2 or 3 seconds to move or to do something. That's those kinds of memories you don't want to lose. And that was the problem with propranolol. Propranolol made them forget some things, didn't make them forget other things. The idea was there, but it kind of dulled people. Since there are several major emotions, different neurotransmitters and structures come into play with the amygdala to create memory: adrenergic, opioid, GABAergic, and cholinergic. The amygdala influences organs that are connected to it through neuronal connections, the hippocampus and the caudate nucleus. Propranolol works by taking the emotion out of the memory. The memory still exists, but the details aren't as distinct. The brain as a whole, and of course that was the problem. It made your emotions less intact, less distinct, but it didn't take the memory away. And then the memory would come back, and of course you're not taking propranolol perpetually, so you added emotions to the memory. And the first time you remembered it, you went, why in the world didn't I react to that? And then afterwards you go, wow, I should have reacted to that. And then later you're going, now I'm reacting to that because now the emotions aren't blocked. The brain as a whole shows a loss of weight beginning at age 30. The prefrontal cortex, or the frontal cortex, loses more weight during this decline than any other portion of the brain. The hippocampus also shrinks with aging, being one of the reasons for the elderly to display memory problems. Another area of the brain that degenerates with age is the septal complex that provides input to the hippocampus. Since most of the neurons in the septal complex use acetylcholine as their transmitter, degeneration of the neurons of acetylcholine producing cells lead to memory problems. Alzheimer's disease is such a memory problem. And that is the end. And so I'll talk to you guys, well, maybe. Anyway, I'll be around this summer. And possibly I will adjunct in the fall. but I am not coming back to Sailly. You guys are at 7,000 feet. I'm at 700 feet. A little bit easier to breathe down here. And my heart doesn't have to work so hard. So talk to you later.